Oh, so thanks a lot, Rui, for this kind introduction, and thanks to Julia and all the organizers for inviting me to give this talk. And I really appreciate this opportunity to share some results that we had with my student. So most of the works that I will be presented today comes from the PhD project of Florian Benitier, and also with a collaboration with Anna Maria Nechulea. And yes, so I will talk about random genetic drift and alternative splicing. And so I was asked to give a broad introduction. So I will start by just describing what is a typical eukaryotic protein coding gene. And as you know, it consists of exons and introns, and these genes are transcribed in the nucleus to give the primary transcript that then gets matured to produce the messenger RNA that can be exported in the cytoplasm to be translated into proteins. And so the maturation of primary transcripts involves the excision of introns, so the process of splicing. And this excision of introns requires several signals, so-called splice signals, located at the extremities of introns and also inside. And one very interesting feature is that thanks to this process of splicing, one single gene can lead to the production of different proteins. And this is possible through the use of the differential use of splice sites during the maturation of the primary transcript. So you can have several molecules of primary transcripts that are spliced in a way to retain, for instance, this exon here, while some other primary transcript will be spliced differently, retaining, for instance, here this green exon. So with one gene, through this process of alternative splicing, the differential use of splice sites, one single gene can express different proteins. And this process of alternative splicing is very pervasive in eukaryotes. And for instance, in humans, it has been shown that something like 95% of multi-exonic genes are subject to this process of alternative splicing. And one interesting observation that was made several years ago is that there is a strong correlation between the rate of alternative splicing and the complexity of organisms. So in that study, Chen and colleagues measured the rate of alternative splicing in more than 100 eukaryotes from 24 different clades. And as a proxy of organism complexities, they measured the number of different cell types in an organism. And what they observed is this very strong correlation between the rate of alternative splicing, the average rate of alternative splicing across genes in the genome, and the number of cell types in the organism. So more complex organisms show a higher rate of alternative splicing. This correlation, however, has to be interpreted with caution because of the issue of phylogenetic inertia. Because here it's just another figure from their paper where they represent their result a different way. So here you have the phylogeny of the taxa that they analyzed. So here you have vertebrates, different invertebrates, fungi, plants, and other unicellular eukaryotes. And as you can, and here what is plotted is the prevalence of alternative splicing in these different species. And so what you can see here is that this nice correlation is essentially driven by the fact that vertebrates, which have a a high diversity of cell types also have a high rate of alternative splicing. So in fact, most of this correlation is driven by the fact that vertebrates have more alternative splicing than other eukaryotes. So still, it's an interesting observation, and we can think about, ask ourselves, what is the raison d'être of alternative splicing? Why do species, why do genomes express these splice variants? And one first explanation is that one first reason why alternative splicing is present is that it contributes to increase the functional repertoire of genomes. And this has been clearly demonstrated. There are some very nice examples of genes that can produce different protein isoforms with different functions through this process of alternative splicing. However, this does not imply that all splice variants are functional, because splicing, like any biological process, is not 100% accurate. In fact, when you sequence a transcriptome, you detect splice variants, and a fraction of them correspond to errors of the splicing machinery. And when I say errors, I mean that these transcripts are costly for the organism, and they are costly because they represent a waste of resources, but also because they can have direct negative effects. Generally, splicing errors lead to frame shifts, and therefore this leads to the apparition of premature termination codon. And so if these transcripts are translated, this leads to, yes, and one clear evidence that these splicing errors exist and are costly for the organism is that, in fact, within cells, there are several cellular machineries that are responsible for detecting and degrading these aberrant mRNA. For instance, there is one machinery that is called the nonsense-mediated RNA decay, NMD, and this machinery is able to recognize mRNAs that contain premature termination codons and to degrade them so that to prevent the expression of aberrant proteins. So the existence of these mRNA surveillance mechanisms demonstrates the importance of getting rid of these erroneous transcripts. And so the question is that, okay, we observe some variation in the rate of alternative splicing across taxa. Does that reflect variation in the repertoire of functional isoforms, or does it reflect simply variation in splicing error rates? And quite a lot of work has already been done, notably to investigate alternative splicing in mammals and notably in humans. And it has been shown that in humans, among all the splice variants that have been annotated in the genome, less than 1% lead to the production of a substantial amount of proteins. So when you look at proteomic data, you observe very few splice variants that lead to a detectable amount of proteins. And in fact, several independent studies led to the conclusion that the majority, more than 70% of splice variants in humans are due to errors of the splicing machinery. And so what determines the splicing efficiency of a given intron? It is known that there are 4 sites that are strictly required for splicing, and essentially this corresponds to the first 2 and the last 2 bases of introns. That in the more than 99% of introns, they start with GT and end with AG. But in addition to these 4 sites, there are about 25 other positions that contribute to the recognition of splice sites. And so suboptimal splice signals lead to decrease the splicing efficiency and therefore to increase the error rate. And what is expected based on population genetic principle is that the quality of splice signal is expected to reflect the balance between mutation, selection, and drift. So on one side, there is selection that will favor the fixation of optimal alleles, but in the other direction, mutation and genetic drift can lead to the fixation of suboptimal alleles. And in species where the effective population size is small, then the intensity of genetic drift is large, which leads to decrease the efficiency of selection. And therefore, it is expected that this should lead to lower the quality of splice signals. And so we might expect, based on population genetics theories, that the splicing error rate should be higher in species with low NE. So we expect a negative correlation between the effective population size, NE, and the splice error rate. So this model derived directly from the nearly neutral theory that was developed by Tomoko Ota. And this theory states that the capacity of selection to purge weakly deleterious mutations depend on the intensity of random genetic drift. And more recently, Mike Lynch popularized these ideas by reformulated these ideas by setting that in fact, the effective population size imposes a barrier to the optimization of genetic systems, so what he calls the drift barrier hypothesis. And so this drift barrier is expected to have an impact on genome evolution, and there is some evidence that various features, genomic features, can be affected by this drift barrier, such as the genome size or mutation rate. And so if I come back to this observation that I mentioned before, this strong correlation between the rate of alternative splicing and the complexity of organism, in fact, there are two possible interpretations. One selectionist interpretation, and which was proposed by the authors of this paper, that in fact, alternative splicing contributes to the evolution of complex organisms by increasing their functional repertoire, or a neutralist interpretation that because in fact complex organisms correspond to large animals, and large animals implies that they have smaller effective population sizes, and therefore more random genetic drift, less efficient selection, so they are more subject to the drift barrier. And therefore, we might expect a higher splice error rate in those complex organisms. So there are these two possible explanations, and the project of Laurent was to test the drift barrier hypothesis, try to investigate whether the rate of alternative splicing correlates with NE across metazoans. And so to test this hypothesis, he investigated the relationship between the rate of alternative splicing and effective population sizes. And as a model organism, we decided to focus on animals, so metazoans, because they cover a wide range of effective population size. So for this analysis, what do we need? We need a reference genome assembly, we need gene annotations, and we need transcriptomic data. Okay, so reference genome, gene annotation, transcriptomic data. And so we were able to investigate alternative splicing in 53 species, so 38 insects and 15 vertebrates. So we focused on these two groups because when we started these studies, this was the two clades for which we had the largest sample sizes. And in total, we analyzed almost 3,500 RNAseq samples, and with an average of 66 RNAseq sample per species. So the first step consisted in aligning all. So for each species, we aligned all RNAseq data against the reference genome to detect splicing events. And we considered all the introns that could be detected, be they annotated or not. And then for each detected intron, we counted the number of reads that cover these introns, and we counted the number of spliced reads that correspond precisely to this intron. So we noticed that N1. The number of splice variants, so splicing events that share one boundary with the focal intron, and counted the number of reads corresponding to non-spliced unspliced reads. So we did this for every single intron that was detected. And in the end, we looked at we measured for each intron the proportion of spliced reads, so N1 divided by N1 plus N2. And so here you have a picture to remind you what N1 and N2 corresponds to. So we have, so here it's for each intron, we computed the proportion of spliced reads that correspond to, that overlap this intron and that correspond to the precise boundaries of these introns. And we looked then at the distribution of this ratio. And as you can see, there is a clearly bimodal distribution. So here it's the example of what we observed for human introns, but we observed very similar picture for all species. And this bimodal distribution is consistent with the fact that for most genes, in fact, there is one single major transcript. And so, in fact, among all introns that we detected, we can distinguish two classes. So the major introns, so those ones that are spliced in the majority of the transcript expressed by a given gene, and the minor splice variants, okay, that are spliced in only a minority of transcript of the transcript of a given gene. So in the end, so for each intron, we compute the rate of alternative splicing, so N2 divided by N1 plus N2. So and so we compute the rate of alternative splicing relative to the major introns for each gene. And to measure the rate of alternative splicing, we only measure it for introns that are covered by at least 10 spliced reads. And we focus on introns that are located within coding regions. So we excluded introns from UTRs or from long non-coding RNAs. One thing that has already been shown in previous studies is that the rate of alternative splicing varies among genes in a genome. And notably, what we had found in a previous work is that the rate of alternative splicing per intron varies according to the level of expression of genes and also to the number of introns in the genes. So this is a study that was conducted in a unicellular eukaryotes here in Paramecium. What is shown here is the rate of alternative splicing according. So what we did is that we binned the genes according to the expression level of the genes well, we bind the introns according to the expression level of the genes in which they are located. And we computed the average rate of alternative splicing per bin. And as you can see, introns located in highly expressed genes show a much lower rate of alternative splicing than introns located in weakly expressed genes. So the rate of alternative splicing varies among genes. And so if we want to compare the rate of alternative splicing across species, it is necessary to analyze the same set of genes. And so for this, we focused on the data set of Busco genes. So this corresponds to orthologous genes that are present in a single copy in most species. And for metazoans, the Busco dataset contains almost 1,000 genes. And so for RNAseq data, essentially what we did is that for every species, we took all the data that were available. I mean, most of the species that we analyzed do not correspond to model organisms. And so for most of them, there are not that many RNA-seq data sets. So to increase the sequencing depths and thereby to limit the variance in the estimate of in the measurement of alternative splicing, we combined all RNA-seq data sets that were available for each species. One point that we realized in the first pilot studies is that we observed that the rate of alternative splicing tended to increase with the number of RNA-seq datasets that we included in the analysis. And to illustrate this, I will show an analysis that we did with human data. So in humans, we have a huge amount of data, and we analyzed 180 RNA-seq datasets. And what we did is that we randomly sampled the datasets, and with a number of RNA-seq datasets ranging from 2 to 20 datasets. And so for each sampling, what we did is that we mapped reads on the genome, and we measured the rate of alternative splicing with this sample. And we also measured the median read coverage on the Busco genes that we obtained with these RNA-seq data. And what is shown here on this plot is the average rate of alternative splicing that we measured according to the read coverage, the median read coverage on Busco genes. And as you can see, when we take a limited number, when we sample a small number of RNA-seq data set, so we have a limited coverage, and as you can see, we get a lower estimate of IS rates. And the more data sets we compile, then we see that the estimate of IS rates converge, reaches a plateau. And the explanation for this is that to measure the rate of alternative splicing, we focused on introns that are covered by at least 10 reads. And so the introns that come from genes that are lowly expressed, they can be included in the analysis only if we have a very high coverage sequencing. And the second point is that, as I mentioned previously, the rate of alternative splicing correlates negatively with gene expression level, so that lowly expressed genes tend to have a higher rate of alternative splicing. And so the average rate of alternative splicing tends to increase with increasing sequencing depth. So the more because when the sequencing depth is limited, we exclude lowly expressed genes in which the rate of alternative splicing is high. So to avoid this artifact, it turned out that it's necessary to focus on species for which we really have a very deep sequencing. So we set a threshold at a median sequencing depth on Busco genes that is larger than 200. So we only retain the species for which the RNAseq datasets that we had allowed to obtain a median read coverage on Busco genes that is larger than 200. And so this is what limited the number of species that we could analyze. Okay, so we have measures of the alternative splicing rate. We need also now to estimate the effective population size. And so we cannot, for most of the species that we analyze, we do not have any estimate of the level of polymorphism. So the only proxies that we could use to for the effective population size were life history traits. So we used longevity and body size as proxies for population for NE for the effective population size, because it is known that long-lived species and large animals tend to have a lower effective population size. And we also, as another proxy of the effective population size, we use molecular evolutionary rate. So we use the DN over DS ratio. So the ratio of non-synonymous over synonymous substitution rate, because it has also been shown that this ratio can be indicative of the intensity of selection, so of the efficacy of selection, and so an indirect estimate of the effective population size. And yes, and here it's just to show that these different proxies of effective population size are correlated between each other. So here it's a correlation between DN over DS and longevity across the 53 species that we analyzed. So in the end, for each of the 53 species, we have the average rate of alternative splicing measured on all Busco genes. So for each introns of Busco genes, and we have 3 proxies of the effective population size. And so we can now test whether there is a correlation between these different variables as predicted by the drift barrier hypothesis. So here is the first result. So here, as a proxy for longevity, for effective population size, we use longevity. So this is an inverse proxy of any. So short-lived species generally have a high effective population size, while long-lived species have a low effective population size. And on the y-axis, it's the average rate of alternative splicing per introns on the introns from Busco genes. And as you can see, there is a variance in the rate of alternative splicing across species, and there is a significant correlation between these two parameters. And we use the PGLS to account for phylogenetic inertia. So even after correcting for phylogenetic inertia, there is a significant correlation between these two parameters. So which implies that there is a higher rate of alternative splicing in species that have a low NE. Yes. And so across overall, there is a 4-fold variation in the rate of alternative splicing. The lowest is observed in Drosophila Grimshawi, and the highest is observed here it's an hymenoptère, and humans here are here with an average rate of alternative splicing of 3.4%, fourfold higher than in Drosophila crimshawi. These are the correlations that we obtain with the two other proxies of effective population size, so body size and DNDS. Again, we see the same correlation, and again, it's significant after accounting for phylogenetic inertia. So the three proxies of any give the same correlation between the rate of alternative splicing and effective population size. So one limitation of our study is that we had to use very heterogeneous RNAseq datasets. So in some species, we have RNAseq data from some particular tissue or organ. In some other species, we have just a whole body extract of RNAseq. Maybe at different life stages, larval or adult. And it is known that the rate of alternative splicing varies across tissues or during development. And so there's a source of variabilities that we cannot control in our data set. And so to evaluate to what extent this might affect our results, we focused on a data set that was compiled by the group of Henrik Kessman, where they used the exact same protocol to extract RNAs from 7 organs, homologous organs across 7 vertebrate species. So here really they control that they have the same protocol across different species, and they focus on the same organs. And so with this set of 7 species, we could measure the rate of alternative splicing across different organs. And as you can see, in each species, there is some variability across organs, and notably in many species, the rate of alternative splicing is highest in testes and lowest in liver. Okay, so you see some variation across organs, but still you see that differences across tissues, across species are larger than the variation across tissues in a species. So we still recover this correlation between the rate of alternative splicing and any, even after controlling for variation across tissues. So in the end, we observe that, as predicted by the Drift barrier, the rate of alternative splicing tends to decrease with increasing effective population size. And so these results suggest that the difference in the rate of alternative splicing across species mainly reflect variation in splicing error rates. But as I mentioned in the introduction, we also know that some splice variants are functional. This has been clearly demonstrated. There are some cases of splice variants that do have a function important for the fitness of organisms. And so
okay,
it seems that every 5 minutes it crashes.
That's fine. Yeah.
Okay, so how can we distinguish functional splice variants from the erroneous ones? So for those splice variants that are functional and that produce proteins, we expect them to maintain the reading frame. And also, we expect that functional variants should be relatively abundant compared to splicing errors. So just to check that these predictions make sense, we plotted here the proportion of variants that preserve the reading frame according to their relative abundancy. Okay, and what you can see is that variants that are rare, I mean, relative to the other transcript produced by a given gene, on average, about 33% of them maintain the reading frame, which is what would be expected by chance. But for variants that are more abundant, you see that clearly the proportion of those splice variants that maintain the reading frame increases with their abundancy. And so clearly this implies that we have among the abundant splice variants, so we set the threshold at 5%. So splice variants that represent more than 5% of the transcript of a given gene, those splice variants are enriched in functional splice variant, as demonstrated by the fact that they have a higher proportion of frame-preserving variants. While the rare splice variants, those below this threshold, here we see no evidence for enrichment in frame-preserving variants. So we have these two sets of splice variants, the abundant ones that are enriched in functional variants and the rare ones that are not. Another way to confirm that those abundant splice variants are functional consists in looking for signatures of selection on these splice variants. And for this, we looked at polymorphism data, and notably in Drosophila melanogaster, polymorphism data, genome-wide polymorphism data are available. And so what we did is that we measured the level of polymorphism, so SNP density, at different positions relative to the splice donor and the splice acceptor. So we show here the first 20 base pair within the intron and the last 20 base pair within the intron. And in the 20 base pair upstream, in the upstream exon or the downstream exon. And so on the y-axis, you have the SNP density. So in the coding region, we distinguish the SNP density at third codon position in yellow, or first or second codon position in gray and red. And the positions within the introns are indicated in blue, and the splice donor, so GT, and splice acceptor, AG, are indicated in dark blue. And as you can see, the level of polymorphism is very low at splice donor and splice acceptor, which simply indicates that these sites are under very strong purifying selection. So most mutations at these two sites are strongly counter-selected. And so we measured the SNP density around on the minor splice sites of splice variants. So what is shown here? So in green, this corresponds to the splice sites of the major introns. And in red, this corresponds to the splice sites of the minor intron, of the alternative splice boundary. And so we measured the level of polymorphism for minor splice sites located within introns or within exon. And as a negative control, we also looked at AG dinucleotides that cannot correspond to acceptor site, or GT dinucleotides that cannot correspond to donor site because they are located here at the 5' end of exons, towards the 5' end of exons, or towards the 3' end of exons. And as you can see, for rare splice variants, the level of polymorphism observed at minor splice sites is much higher than what is observed for the major splice sites. And it's not very different from what is observed at control regions. So not a strong evidence. We see here some evidence for a selective pressure on minor splice sites within introns, but within exons, it's not detectable. So this is what we observe for rare splice variants. But now when we looked at the abundant splice variants, we see a very different picture with a clear deficit of polymorphism on minor splice sites, both located within those located within introns or those located within exons, as compared to control sites. So this is clear evidence that on abundant splice variants, the splice sites are indeed under selective pressure, which is good evidence that these splice variants are enriched. These abundant splice variants are enriched in functional splice variants. Okay, so now, okay, we have rare splice variants in which we see little evidence for functional constraints. And when we look at the correlation between the rate of alternative splicing in these splice variants and NE, so here we took longevity as a proxy for NE, we do see this this clear correlation that we showed previously. And this is expected because these rare splice variants correspond to 90 to 95% of I mean, these introns that have only rare splice variants, they correspond to 90 to 95% of all introns. And for those major introns that have some abundant splice variants, here we see a very different picture. Here we see that the rate of alternative splicing tends to decrease with increasing longevity. So we see a pattern that goes in the opposite direction to what we see for introns that have only rare splice variants. So those introns with rare splice variants, we see that the rate of alternative splicing increases when the effective population size decreases. So it's consistent with the drift barrier hypothesis. So a higher rate of splice error in species with low NE. While for major introns with abundant splice variants, so those ones that are enriched in functional splice variants, we see the opposite trend. So the rate of alternative splicing tends to increase when the effective population size increases. And so, if anything, it shows that the more complex species, they do not have more functional alternative splicing. If anything, it goes the opposite direction. An additional prediction of the drift pair model is that the rate of alternative splicing is also expected to vary among genes in a genome. Because so the rate of alternative splicing, in fact, corresponds to a combination of the rate of splice error plus the rate of functional splice variance. And for the errors, we know that the increase is the cost of errors increases with the expression level, because the higher the level of gene expression, the higher the waste of resources each time you have errors, splicing errors. So you expect highly expressed genes to be under stronger selective pressure to minimize splicing errors. And so you expect a lower rate of splicing errors among highly expressed genes. So the drift barrier prediction is that there should be a negative correlation between splice error rate and gene expression level across genes in a genome. And so these introns that do not have abundant splice variants, so those that only have rare splice variants, which represent the vast majority of introns, we don't see any evidence of functionalities in these splice variants, and most of them probably correspond to errors. And therefore, what we expect is that there should be a negative correlation between the rate of alternative splicing and expression level at those introns. And so we check that, and this is, for instance, what we observe in humans. So what we did is that we binned introns according to the expression level of their gene. And for each bin, we compute the average rate of alternative splicing. And here in human, you can see that, yes, there is a negative correlation between the rate of alternative splicing per intron and the level of gene expression. In Drosophila, the pattern is less clear, but still there is this negative correlation, significant negative correlation between gene expression level and the rate of alternative splicing. And so we computed this correlation for all species. And in fact, out of 53 species, 52, all but one, showed a negative correlation. And this correlation was significant in 49 of the 53 species. So this negative correlation between the rate of alternative splicing and gene expression is observed in the vast majority of species. So this is consistent with the drift barrier model, and this is thus consistent with the hypothesis that for most introns, splice variants correspond to splicing errors. And so our model to explain this pattern is the following. So what we think is that, in fact, the rate of alternative splicing errors, the rate of splicing errors varies among species according to their effective population size. So in species with high effective population size, the rate of splicing error is lower than in species with low effective population size. And we assume that a small fraction of introns undergo functional splicing, alternative splicing. Okay, and so just to illustrate, for the purpose of illustration, imagine that, let's say, 5% of introns undergo functional alternative splicing. And let us assume that this rate of functional alternative splicing is the same across all species, with a rate of alternative splicing for these functional variants at an average of 25%. So this is a small bump that you can barely see here. So I just zoom on the distribution so that you can clearly see. So what we assume is that, okay, in all species, a small fraction of introns undergo functional alternative splicing, and with the same rate across species. And what is changing across species is simply the rate of splicing errors. And overall, when you measure the total genome-wide rate of alternative splicing, so it's a combination of functional splice variants and splicing errors, you see that this rate varies according to NE. And so these are the prediction of the model, of this mixture model. You expect that the average rate of alternative splicing increases when in species in low NE compared to high NE. And this is what we observed. So it's consistent with the prediction. And what the model predicts is that this trend should be observed for introns with rare splice variants, but we expect the opposite trend for introns with abundant splice variant, and this is indeed what we observe. One last point to finish. I mentioned before that in Drosophila, we see evidence of selection on of selective pressure on the minor splice sites for the abundant splice variants. So this is what we observed in Drosophila. We looked at the same thing in humans, but in humans, even for the abundant splice variants, we see no evidence of selective pressure on the minor splice sites, which implies that even the abundant splice variants in humans, a vast majority of them show no evidence of functionality. So to conclude, splice variants correspond to a mixture of functional variants and splicing errors. And only a small minority of splice variants are functional, and typically those ones are frame preserving, and they are relatively abundant, and they have splice sites that are under purifying selection. And in fact, variation in the rate of alternative splicing across species and within genomes fit very well with the drift barrier hypothesis, so that the differences in average alternative splicing rate across species mainly reflect variation in splicing error rates and not a higher prevalence of functional splice variants in complex species. And so our conclusion is that random genetic drift sets an upper limit on splicing accuracy. And I realize that I'm a bit long, and so I will just quickly go through the conclusion that the drift barrier hypothesis, I think, is a very general model, and it's a very insightful model, but in the end, there is not that much empirical support. There is evidence that the rate of mutation increases with decreasing NE. We show here that it's also true for the rate of alternative splicing. For genome size, it's not so clear. There are some studies that say yes and others that say no. And for the transcription error rate, it's not so clear. There's no it does not fit with the drift barrier prediction of the drift barrier hypothesis. So I think it's an interesting question to investigate to what extent random genetic drift contribute to variation in genomic architecture and in gene expression across the tree of life. And with that, I will thank you for your attention, and I'd be happy to take questions. And sorry for being a bit long.